Hey everyone, in this video I am going to talk about HIV-1 integration into the genome. So the genome of its host cell. And as you saw in previous videos, this is my diagram of an infectious virion, or infectious particle of HIV-1. And HIV-1 is what we would call a single-stranded RNA virus, and each infectious particle has 2 copies of the genome, but I only diagrammed one here. And the goal of HIV-1 is to get this copy of this single-stranded RNA in DNA form into the genome of the cell it infects. And if it can do that, well, then that cell can make more HIV virions that can be released from the cell and they can go infect other cells that haven't been infected yet. So in this video, I'm going to talk about how that DNA copy of the HIV-1 genome is integrated into the genome of a host cell. So in the previous video, I talked about how the single-stranded RNA was turned into a DNA that could be integrated into the genome. And so we're short on time, so I'm not going to do that much diagramming of the notes. I'm just going to go through some pre-prepared notes that I put together so we can move through this video rather quickly. So I'm going with Wikipedia's 6-step procedure for integration of HIV-1 into the genome, or the 6-step mechanism. So step 1 is going to be binding of the integrase. So you saw in a previous video that HIV makes at least 15 different proteins. One of the proteins was integrase. And here I have 4 copies of the integrase that are bound to each to the LTRs, so the long terminal repeats of the HIV-1 DNA. So this is after reverse transcription, and that after the RNA has been turned into a DNA and the LTRs have been restored to the HIV-1 DNA. So step 2 is going to be 3' end processing. So those integrases are thought to remove 2 nucleotides from the ends of the HIV-1 DNA. So that leaves a 2 nucleotide 5' overhang on each end. So and these sequences are conserved, and it's thought that the GT 5' GT dinucleotide is released on each end. So essentially it's leaving a 5' AC dinucleotide on each end. And that's it. So just a really simple 3' end processing on each end of the HIV-1 DNA. Step 3 is going to be translocation to the nucleus. So this is rather interesting. So all that reverse transcription, binding of the integrase to the ends of the LTRs of the HIV-1 DNA, processing of the ends, all of that is thought to occur inside that capsid. So if you remember from our structure of HIV-1, we have the matrix protein, which is right under the envelope, viral envelope, but inside there we have the capsid, which encases or encloses the RNA. Now, inside that capsid, that's where we think reverse transcription is going on. The RNA is being turned into a DNA, and the integrases, after the DNA is formed, are processing the ends. So all of that leads to this step right here. All of that is thought to occur in the cytoplasm of the host cell. And this whole capsid here, it's thought to be able to travel through the nuclear pore. And once inside the nucleus, well, then the capsid can dissolve or fall apart and release the HIV-1 DNA with the 3' process ends into the nucleoplasm, which is what we call the stuff that's inside the nucleus. So once in the nucleus and once outside of that capsid, well, we can have tethering. Step 4 is tethering of the HIV DNA with the 3' processed ends to the host DNA. And a host protein is thought to be really important for that process. That host protein has a funny name, LEDGF-P75. So this host protein, and viruses are really neat in their ability to do that, use our own proteins against us. So this protein right here is thought to tether the HIV-1 DNA to a location in the host genome. And I'm not implying here that this is a preferred sequence for HIV-1 integration, but I wanted to just use a sequence that would allow me to show how the target site duplication works with respect to HIV integration. So I made up a sequence with a bunch of A's on either side of a bunch of C's. And of course the complementary strand is down here with the T's and G's. So we have tethering of the HIV-1 DNA to host DNA, and you can see I have this little circle here. So it's thought that those integrases, those integrase dimers, come together to form like a tetramer. So we have this integrase tetramer here, and we've got these ends rather close together. So these are the 3' processed ends of the HIV-1 DNA. Now, it's thought that the OH groups on these 3' ends are used to break bonds and cause a double strand break in a staggered double strand break, breaking a bond here and another one 5 base pairs away on either side of the host DNA. And those 3' OH groups are going to be ligated. To the nucleotides here to help integrate this DNA into the host DNA, to help integrate the HIV-1 DNA into the host DNA. So that's step 5, integration. You're breaking these bonds here and ligating the HIV-1 into the host DNA. So let's see, what will that look like? It's kind of hard to visualize how this happens, especially with a 2-dimensional diagram. So it will look something like this. So that 3' right here, I'm going to show you this 3' end I'm going to put here, and this 3' end I'm going to put here. So that's going to give us something that looks like this. So we have this that staggered double strand break. Oh, no, I'm sorry. That occurred where I put those arrows and the ligating of the 3' ends to the places where the double strand breaks were formed. And now we have these gaps, right? So these are linked right here with a covalent bond. These are linked right here. However, we remember those 5' overhang of the HIV-1 DNA? Well, those might not necessarily match the host DNA. So step 6 is to fill the gaps, ligate any nicks, and either fix the mismatches or remove these flaps. And the flaps, what am I talking about? That's the 5' overhang. Remember, there were 5' overhangs on that DNA molecule after 3' end processing. So here's a 5' overhang and a 5' overhang. So these may not match the host DNA after the 3' ends are ligated into the host DNA. And in this case, only the C matches, the A doesn't. And over here, neither the CA overhang doesn't match the CC of the host. So these are going to have to be removed by some sort of nuclease, or they're going to have to be fixed by mismatch repair. But as we've seen in many of our other mechanisms, when we have gaps, what do we need? We need a DNA polymerase. This is all going to be host. These are probably all going to be host enzymes. DNA polymerase is going to come in, fill that gap, and seal any nicks that remain. Same thing for this end over here. And if there are any mismatches behind, well, then mismatch repair or some DNA repair process like base excision repair or something is going to have to come in and fix these. There are a lot of researchers that think that these just leave flaps, and those flaps are removed by some sort of endonuclease. In either way, any mismatches here are going to have to be repaired. After all of that, what we're going to have is the HIV-1 provirus inserted into the genome of the host, and we have our target site duplication, right? So let's go back a little bit. So we had the breaks on either side of this 5 base pair sequence, which is flanked by A's and T's. See, after it integrates, we have the target site was duplicated on either side of the HIV-1 provirus integration site. Okay, so that's it. That's how HIV-1 is thought to, at least that's one of the current models of how HIV-1 integrates into the genome of a host cell. And I think after these 3 videos, we should be ready to understand how HIV-1 has been used to develop viral vectors for gene therapy in humans. So, okay, so see you in the next video when I will introduce HIV-1 based viral vectors.